Hi, I'm Pamela Horton, and this past summer, under the guidance of Dr. Healy, I attended a summer research program at Texas A&M Health Science Center in College Station. Although I'm a chemistry major, Dr. Healy encouraged me to branch out a little and try some more biology-focused research because I'm pre-med and planning on attending medical school this summer. While I was at the research program, I was a part of the Muthachami lab and got to do some research with animal models, which was something really cool that I had never done before. Dr. Muthachami's main research focus is the lymphatic system and the mechanics of lymphatic flow, but this project was actually a collaboration between his lab and Dr. Reddy's lab in the neuroscience department. The goal of this project was to study how the lymphatic system around the brain remodels after induced seizures in mice. Nearly 50 million people worldwide suffer from epilepsy in some form. Epilepsy may be caused by infection, brain damage, fever, or it may have no obvious cause. I'm sure most of you have seen some version of epileptic seizures in TV or movies, but in reality, there are a lot of different kinds of seizures with different symptoms and appearances. Focal onset seizures are limited to one hemisphere of the brain, while generalized onset seizures occur in both hemispheres. Focal onset seizures are further divided into seizures where the person is aware and they can react to their environment, and seizures where they have impaired awareness. That may mean that they have amnesia afterwards, or they're completely unconscious. Now, these different kinds of seizures, aware and impaired awareness, are also divided into motor onset and non-motor onset seizures. Motor onset seizures are the seizures that include the kind that you might see on TV where people are thrashing around. Those tonic-clonic seizures, previously known as grand mal seizures, are not the only type of motor seizure, however, and the degree of thrashing around or twitching may be different depending on the severity of the seizure. Non-motor onset seizures include other senses of the body like taste or smell. The person may have hallucinations, or they may have neurological symptoms like aphasia or forgetting words. Finally, there are unknown onset seizures. Now, like focal onset and generalized onset seizures, these seizures are divided into motor and non-motor. However, not enough is known about the specific seizure to be able to classify it as single hemisphere or dual hemisphere. Epilepsy is a disorder characterized by spontaneous recurrent abnormal electrical signals in the brain. Typically, epileptogenesis, or the onset of epilepsy, follows this pattern where the individual suffers an initial epileptogenic injury, like a traumatic brain injury. This injury causes some pathology like cell death due to too many excitatory signals, alteration of proteins in the neural synapse, or generalized neural inflammation. The initial injury is followed by a latent period of a few months or even a few years in humans, wherein the neurons restructure and the synapses reorganize. This can lead to an imbalance or a disability to regulate excitatory signals in the brain. The cells can even train themselves to all react together or react in synchronized activity, which may lead to a positive feedback loop of excitatory signals, which is toxic to the cells and can lead to death. Chronic epilepsy emerges as a result of this latent period restructuring and is characterized by recurrent spontaneous seizures. As you may have noticed, one of the potential causes of initial injury is status epilepticus, or SE. SE is characterized by a continuous seizure lasting more than 30 minutes or by multiple seizures without recovery in between. This is a life-threatening condition and can lead to brain damage or even death. The mortality rate of SE varies from 3 to 50%, depending on the age of the individual and the time it takes for the sufferer to receive treatment. As I'm sure you all have noticed by now, my presentation has been very neurology focused so far. So you may be wondering what this has to do with cardiovascular research or the lymphatic system. Scientists and medical researchers have assumed for decades that the central nervous system lacks lymphatic drainage because we previously did not know that there were lymphatic vessels surrounding the brain. In 2015, these lymphatic vessels were discovered in the dura mater. As you can see from this diagram, the meningeal layers surrounding the brain are the pia mater, the layer closest to the cerebrum, the arachnoid mater, and the dura mater. The dura mater is the hardest layer and contains the sinuses. Because this discovery is so new, very little is known about the lymphatic drainage system surrounding the brain. It has been known for some time that the cerebrospinal fluid, or CSF, that supports the brain brings nutrients to neurons and their supporting glial cells and eliminates metabolic waste products. And this system drains to the lymphatic system. But the discovery of these lymphatic vessels are kind of the missing link that really confirm that suspicion. So the lymphatic system itself is quite mysterious, and research in the lymphatic system is not nearly as advanced as research in the blood vessel system. But the lymphatic system serves a variety of functions in the body, like carrying digested fats in the form of chylomicrons and draining fluids from tissues suffering swelling. Disruptions in the lymphatic system can lead to gross swelling called lymphedema. Because we don't know very much about the lymphatic system, lymphedema is treatable but incurable. So clearly, even minor disruptions to the lymphatic system balance, the balance between fluid leaking out of the capillaries and being reabsorbed from the interstitial tissues, can have dramatic consequences. These disruptions can be caused by injury and disease. Lymphatic vessels must therefore be able to regulate themselves. In the instance of injury, lymph vessels must proliferate and increase in the complexity of their branching in order to absorb more fluid from the interstitial spaces and from the tissues to prevent swelling. They also must be able to drain the inflammatory markers from inflamed tissues to avoid something like the cytokine storm that we're seeing in COVID infections. So we know that in epilepsy and after status epilepticus, after any kind of neuronal injury, the blood vessels around the brain undergo angiogenesis, or blood vessel formation. So we have hypothesized that if angiogenesis occurs, and if there are lymphatic vessels surrounding the brain, then the lymphatic vessels must also multiply, a process called lymphangiogenesis, and they must restructure and increase in branching and complexity as a result of status epilepticus. This would serve to help drain the area and prevent uncontrolled swelling around the brain, as well as remove pro-inflammatory signals from the area during recovery. We also hypothesized that in analysis of the lymphatic drainage through the cervical lymph nodes, we might see an increase in inflammatory markers and immune cells. This would represent the clearance of cells from the initial acute injury-associated flood of cells. So one of the ways that people study epilepsy is by using animal models. And one of the ways of inducing epilepsy in these animals is by using pharmacologic stimulation. Injection with a drug like pilocarpine can mimic the initial neurological injury in the epileptogenesis model. In mice, pilocarpine injection is typically followed by 24 hours of status epilepticus, then a latent period of at least a month, followed by the onset of chronic epilepsy. Pilocarpine is a muscarinic agonist, meaning it binds to muscarinic acetylcholine receptors in the nervous system, causing muscle contractions. In our study, we primed the mice first with an injection of scopolamine prior to the pilocarpine injection. Scopolamine is an anticholinergic drug which binds to acetylcholine receptors and reduces the amount of pilocarpine that we need to inject, thereby improving mouse prognosis. After approximately 90 minutes of status epilepticus, we injected the mice with diazepam, a drug used to treat epilepsy, in order to control the status epilepticus and prevent mortality. We collected samples from mice after 24 hours of status epilepticus and at 72 hours, which was during that latent period. These were compared with samples from our control mice, which were not injected with pilocarpine. Our goal was to see how the lymphatic structure rearranged in the meninges and how lymphatic flow through the cervical lymph nodes was affected by the status epilepticus. So one of the ways that we wanted to visualize these structural changes in the lymphatic vessels in the meninges was to use immunohistochemical staining. So in this process, at the time of collection, we extracted cerebral samples, attempting to preserve the meninges surrounding the cerebrum, and dyed it with a marker that's specific for lymphatic endothelial cells, LIVE1. LIVE1 stands for lymphatic vessel endothelial cell receptor 1 and is relatively specific for lymphatic endothelial cells. So the process by which we did this was we froze our samples initially, and then we sliced them into 30 micron sections using a cryostat. We fixed the tissues with paraformaldehyde and stained them with a fluorescent dye. We first used a primary antibody which bound the LIVE1 positive cells in our samples. And then after incubation with that solution, we used a secondary antibody containing the fluorescent molecule, which bound to our primary antibody and allowed us to visualize these LIVE1 positive cells. To quantify our results, we also used a DAPI stain, which stands for individual nuclei. And with that ability to visualize individual cells, we counted all of the LIVE1 positive cells in the superior half of each hemisphere of the cerebrum. So here's kind of an example of what we saw. So all of these little blue dots are individual nuclei, and everything green is the LIVE1 positive regions. So in the quantification, we just counted all of the little blue dots in the LIVE1 positive regions in the kind of top half of the brain. So this is kind of a quantification of the cell counts that we obtained. And I don't know if you can see the asterisk, but we did find a statistical increase in LIVE1 positive cells between the control and the 24-hour time point and the control and the 72-hour time point. So we believe that this increase in LIVE1 positive cell density indicates lymphatic endothelial cell proliferation or lymphangiogenesis. This would confirm or support our initial hypothesis that lymphatic cells proliferate to control swelling in inflammatory cell populations around the neural injury. But it is important to note that these results are preliminary, and because of our small sample size, we did have a relatively large error. In the future, we would like to repeat this same kind of study using a larger sample size in order to reduce variance and increase significance. And we would also like to try the same kind of study, but using a counterstain of protoplanin. Although LIVE1 is relatively specific for lymphatic endothelial cells, it can also be found in macrophages. So using a different stain that's also specific for lymphatic cells can help us confirm that the cells that we saw are in fact lymphatic in origin. And using a counterstain may also help us actually trace the lymphatic vessels in the meninges. One of the challenges in this part of the study was extracting the cerebra and preserving the meninges. This isn't something that either labs have really tried before. So it was really tricky to kind of peel off the skull. And I'd be interested to see what would happen if we tried to use the cryostat without peeling off the skull. Although that may be more expensive in terms of cryostat blades. In the future, we also hope to do some live animal studies using tracer molecules and external imaging to kind of visualize the lymphatic flow before and after status epilepticus. So the other kind of experiment that we did over the summer was looking at how the lymphatic flow and the composition of the cerebrospinal fluid that drains actually changes. So in this process, we isolated the cervical lymph nodes at the same time that we isolated the cerebra. We homogenized our samples and purified the RNA. And then we performed reverse transcriptase reactions for specific inflammatory gene primers. And the goal of this was to see how inflammatory genes change in expression before and after status epilepticus. So we kind of thought that once the mice undergo this neuronal injury, immune cells and inflammatory markers will kind of flood to the region and drain down through the cervical lymph nodes. And we analyzed this using real-time quantitative PCR, which allows us to amplify gene expression and compare the relative amounts of expression for the different genes that we're testing for. So this is a lot of data here. So I'm going to stick to the genes that were most significant, but if y'all want to ask me about the other genes that we tested, feel free to do that at the end of the presentation. Okay, so first off, we found an increase in this INOS, as well as IL-1 beta, MCP1, and IL-6 at the 24-hour point, but not at the 72-hour point. So to understand a little bit about what these genes do, IL-1 beta is an interleukin, or a gene that codes for an interleukin, which is a molecule that regulates the immune response. IL-1 beta specifically is responsible for cell regulation, proliferation, and apoptosis, which is programmed cell death, and it is associated with inflammation. IL-6 is another interleukin, which is a pro-inflammatory cytokine associated with acute inflammation. And cytokines are molecules that attract other cells, specifically immune cells, to the region. INOS is inducible nitric oxide synthase, and it produces nitric oxide, which is associated with immune cell function and inflammation. And MCP1 is produced by macrophages in response to immune cell stimulation and is also associated with inflammation. So something that was interesting that we didn't particularly expect was that these would increase at 24 hours and return to baseline at 72 hours. So this kind of indicates to us that these are mainly involved in the acute response to inflammation. And they might return to normal to protect the neural tissue from prolonged inflammation. So in the CC cytokines that we looked at, we saw increases and decreases in CCL19, CCL21, CCR7, CXCL1, and CXCR2. So for reference, the L in the name refers to a ligand, which means it's the molecule that attracts other cells to the location. And the R indicates that it's a receptor, so that molecule is found on cells that are being attracted to a location. So CCL19 and CCL21 are ligands for CCR7, and we found that all of them decreased at the 24-hour time point, but the responses are kind of variable at the 72-hour time point. So CCR7 is located on dendritic cells and lymphocytes, which are more pathogen-targeting immune cells. So we thought that maybe these decreased because there's no pathogen to target in status epilepticus. So maybe these kinds of cells are outcompeted in the location by other immune cells. CXCL1 binds to CXCR2, and we saw that CXCL1 increased drastically at the 24-hour time point. And this ligand is associated with neuroprotection. So we kind of figured that that's kind of obvious in the acute neural response that this would increase to kind of try and protect the neural cells. And then this returned to baseline at the 24-hour time point, while the CXCR increased. So we kind of thought that these might have only increased slightly at the 72-hour time point because they might be slightly involved in inflammation-associated angiogenesis, because CXCL1 is normally associated with angiogenesis in addition to neuroprotection and inflammation. So we definitely saw an alteration in gene expression of inflammatory markers in the CLNs following induction of status epilepticus. Some of the challenges were the small sample size, which I mentioned earlier. And in the future, we definitely would want to compare the lymph nodes from other areas of the body to see if these are kind of overall systemic responses or specific to the cervical lymph nodes and the meningeal drainage. We would also like to sample farther along the timeline of the epileptogenesis model to see how the latent period and the chronic epilepsy period alter the brain chemistry and meningeal structure. So overall, we saw that pilocarpine-induced epilepsy in mice causes an inflammatory response in the brain based on our data regarding the inflammatory marker gene expression. And we also saw an increase in LIVE-1 cell density in the meninges, which provides a basis for further work on how the actual lymphatic structure changes. So we believe that this data is potentially really useful and a good baseline to potentially develop lymphatic-based treatments for epilepsy in the future, and ways to control drainage and preserve neural function after neuronal injury. So I'd like to thank my advisor, Dr. Healy, Dr. Muthuchami for hosting me this summer, and Dr. Wong, Wu, and Reddy for helping me out with providing the mice and actually doing the experiments. So this work was supported by the American Heart Summer Undergraduate Research Fellowship and the Texas A&M Health Science Summer Research Program. Thank you.
Okay, hi everybody, welcome back. Dr. Healy, I'd like to invite you to share a few words about Pamela, and then if anybody wants to put some questions in the chat function, we may be able to take one question, but then we'll be running short on time.
Well, can you hear me? As impressive as this work is, it forms just a small part of Pamela's impressive undergraduate research career. So Pamela started with me, it seems so long ago now, she started with me either as a freshman or as a sophomore, and we did some molecular modeling of HIV integrase. And as we were progressing, I could see that Pamela wanted to spread beyond that. And so as part of our NIH collaboration with the University of Texas, Pam, I think in the summer of her sophomore year, went off to work with Walter Fast, working on a covalent modified inhibition. She did some beautifully elegant work, both synthetic and enzymatic kinetic work. She then came back to St. Edward's, of course, and she started modeling those inhibitors that she had worked with in Dr. Fast's lab. And not surprisingly, found that the modeling results and the experimental results meshed perfectly. And just as I was thinking that maybe we could get in touch with Dr. Fast and mold all of this into a paper, Pamela thought that she hadn't done enough. There wasn't enough kind of clinical outlet for her work. And so she started looking at REUs and obviously ended up with Dr. Reddy and Dr. Machumo at A&M doing absolutely wonderful work with the pilocarpine-induced epilepsy. So at this stage, Pamela is off to UTMB med school. And my guess is that the clinical work and her exposure to the clinical work won't be enough. So if I was them, I'd be well prepared for her knocking on the basic research door. And I really hope that she maintains and expands both her clinical, but also her basic research abilities. Because Pam, I think your future is enormously bright and you have an enormous opportunity to contribute in many ways. And now there is never a better time for somebody with your skills. Thank you, Pam, for all your interaction with me.
Thank you.
Okay, any questions? I haven't received any in the chat box.
Okay. Sorry, I don't want to step on anybody, but I actually do have a question because unfortunately I was going to sit down with Pam and we were going to go through some of the results. Pam, you know my interest is in cytokines. In terms of CXCL1 or CXCR6, they're both constitutive and induced. Are they shed or are they actual membrane attached? Do they remain membrane attached, or can they be shed by the shedases? And so do they have kind of a totally soluble cellular component?
So I believe that CXCL1 is constitutively expressed around the brain because it's required for neurogenesis and myelination. And I know that it's expressed in macrophages as well. So I believe that constitutive expression is, cell bound, but because it's also expressed and released from macrophages, it's also kind of mobile. And those can concentrate in an area and increase the relative expression. Wow.
Yeah, I mean, that is obviously a very complicated interplay right there. And the amount we don't know about these cytokines is absolutely enormous. Thank you, Pam.
Okay, unfortunately, we have to move on to our next presentation, which is Sam Kenyon, and we're going to get started. He is present as well as his advisor. So I will be keeping time for you guys. And at the end, if you have any questions, we'll take them through chat or live. So I'm going to start your video now, Sam.
Gotcha.